Natural compound found in broccoli reawakens the function of potent tumor suppressor. Your mother was right, broccoli is good for you. Long associated with decreased risk of cancer, broccoli and other cruciferous vegetables, the family of plants that also includes cauliflower, cabbage, collard greens, Brussels sprouts and kale, contain a molecule that inactivates a gene known to play a role in a variety of common human cancers. In a new paper published today in Science, researchers, led by Pier Paolo Pandolfi, MD, PhD, director of the Cancer Center and Cancer Research Institute at Beth Israel Deaconess Medical Center, demonstrate that targeting the gene, known as WWP1, with the ingredient found in broccoli suppressed tumor growth in cancer-prone lab animals. We found a new important player that drives a pathway critical to the development of cancer, an enzyme that can be inhibited with a natural compound found in broccoli and other cruciferous vegetables, said Pandolfi. This pathway emerges not only as a regulator for tumor growth control, but also as an Achilles heel we can target with therapeutic options. A well-known and potent tumor-suppressive gene, PTEN is one of the most frequently mutated, deleted, down-regulated or silenced tumor suppressor genes in human cancers. Certain inherited PTEN mutations can cause syndromes characterized by cancer susceptibility and developmental defects. But because complete loss of the gene triggers an irreversible and potent failsafe mechanism that halts proliferation of cancer cells, both copies of the gene, humans have 2 copies of each gene, one from each parent, are rarely affected. Instead, tumor cells exhibit lower levels of PTEN, raising the question whether restoring PTEN activity to normal levels in the cancer setting can unleash the gene's tumor suppressive activity. To find out, Pandolfi and colleagues identified the molecules and compounds regulating PTEN function and activation. Carrying out a series of experiments in cancer-prone mice and human cells, the team revealed that a gene called WWP1, which is also known to play a role in the development of cancer, produces an enzyme that inhibits PTEN's tumor suppressive activity. How to disable this PTEN kryptonite? By analyzing the enzyme's physical shape, the research team's chemists recognized that a small molecule, formerly named indole 3-carbonyl I3C, an ingredient in broccoli and its relatives, could be the key to quelling the cancer-causing effects of WWP1. When Pandolfi and colleagues tested this idea by administering I3C to cancer-prone lab animals, the scientists found that the naturally occurring ingredient in broccoli inactivated WWP1, releasing the brakes on the PTEN's tumor suppressive power. But don't head to the farmer's market just yet. First author Yu-Ru Lee, PhD, a member of the Pandolfi lab, notes you'd have to eat nearly 6 pounds of Brussels sprouts a day, and uncooked ones at that, to reap their potential anti-cancer benefit. That's why the Pandolfi team is seeking other ways to leverage this new knowledge. The team plans to further study the function of WWP1 with the ultimate goal of developing more potent WWP1 inhibitors. Either genetic or pharmacological inactivation of WWP1 with either CRISPR technology or I3C could restore PTEN function and further unleash its tumor suppressive activity, said Pandolfi. These findings pave the way toward a long-sought tumor suppressor reactivation approach to cancer treatment. In addition to Pandolfi and Lee, authors include Ming Chen, Jonathan D. Lee, Jinfang Zhang, Tomoki Ishikawa, Jesse M. Caton, Yang Zhang, Yulia V. Sholga, Asif C. Bester, Jacqueline Feng, Emmanuel Monteleone, Lizin Wan, John G. Clahisi, and Wenyi Wei, all of BIDMC, Xu Yu Lin, Shang Yin Chang and Rui Hua Chen of Institute of Biological Chemistry, Tian Min Fu and Chen Shen of Harvard Medical School, Chi Hung Shu, Hao Chen and Hao Wu of Boston Children's Hospital, Antonella Papa of Monash University, Julie Teruya Feldstein of Icahn School of Medicine at Mount Sinai, Suresh Jain of Intonation Research Laboratories, and Lydia Matisik of University of South Carolina. This work was supported by the National Institutes of Health R01CA82328 and R35CA197529, granted to Pandolfi. Lee was supported in part by the Postdoctoral Research Abroad Program Fellowship, Taiwan National Science Council (NSC), and DoD Prostate Cancer Research Program (PCRP) Postdoctoral Training Award W81XWH, the 16th of January 249. Disclosures: Pandolfi, Wei and Suresh Jain are co-founders of Rekindle Pharmaceuticals. The company is developing novel therapies for cancer. All other authors declare no competing interests. Anxiety might be alleviated by regulating gut bacteria. People who experience anxiety symptoms might be helped by taking steps to regulate the microorganisms in their gut using probiotic and non-probiotic food and supplements, suggests a review of studies published today in the journal General Psychiatry. Anxiety symptoms are common in people with mental diseases and a variety of physical disorders, especially in disorders that are related to stress. Previous studies have shown that as many as a third of people will be affected by anxiety symptoms during their lifetime. Increasingly, research has indicated that gut microbiota, the trillions of microorganisms in the gut which perform important functions in the immune system and metabolism by providing essential inflammatory mediators, nutrients and vitamins, can help regulate brain function through something called the gut-brain axis. Recent research also suggests that mental disorders could be treated by regulating the intestinal microbiota, but there is no specific evidence to support this. Therefore, a team of researchers from the Shanghai Mental Health Center at Shanghai Jiao Tong University School of Medicine set out to investigate if there was evidence to support improvement of anxiety symptoms by regulating intestinal microbiota. They reviewed 21 studies that had looked at 1,503 people collectively. Of the 21 studies, 14 had chosen probiotics as interventions to regulate intestinal microbiota (IRIFs), and 7 chose non-probiotic ways, such as adjusting daily diets. Probiotics are living organisms found naturally in some foods that are also known as good or friendly bacteria because they fight against harmful bacteria and prevent them from settling in the gut. The researchers found that probiotic supplements in 7 studies within their analysis contained only one kind of probiotic, 2 studies used a product that contained 2 kinds of probiotics, and the supplements used in the other 5 studies included at least 3 kinds. Overall, 11 of the 21 studies showed a positive effect on anxiety symptoms by regulating intestinal microbiota, meaning that more than half, 52%, of the studies showed this approach to be effective, although some studies that had used this approach did not find it worked. Of the 14 studies that had used probiotics as the intervention, more than a third, 36%, found them to be effective in reducing anxiety symptoms, while 6 of the remaining 7 studies that had used non-probiotics as interventions found those to be effective, a 86% rate of effectiveness. Some studies had used both the IRIF interventions to regulate intestinal microbiota approach and treatment as usual. In the 5 studies that used treatment as usual and IRIF as interventions, only studies that had conducted non-probiotic ways got positive results that showed a reduction in anxiety symptoms. Non-probiotic interventions were also more effective in the studies that used IRIF alone. In those studies only using IRIF, 80% were effective when using non-probiotic interventions, while only 45% were found to be effective when using probiotic ways. The authors say one reason that non-probiotic interventions were significantly more effective than probiotic interventions was possible due to the fact that changing diet, a diverse energy source, could have more of an impact on gut bacteria growth than introducing specific types of bacteria in a probiotic supplement. Also, because some studies had involved introducing different types of probiotics, these could have fought against each other to work effectively, and many of the intervention times used might have been too short to significantly increase the abundance of the imported bacteria. Most of the studies did not report serious adverse events, and only 4 studies reported mild adverse effects such as dry mouth and diarrhea. This is an observational study, and as such, cannot establish cause. Indeed, the authors acknowledge some limitations, such as differences in study design, subjects, interventions and measurements, making the data unsuitable for further analysis. Nevertheless, they say the overall quality of the 21 studies included was high. The researchers conclude, "We find that more than half of the studies included showed it was positive to treat anxiety symptoms by regulation of intestinal microbiota." There are 2 kinds of interventions, probiotic and non-probiotic interventions, to regulate intestinal microbiota, and it should be highlighted that the non-probiotic interventions were more effective than the probiotic interventions. More studies are needed to clarify this conclusion since we still cannot run meta-analysis so far. They also suggest that, in addition to the use of psychiatric drugs for treatment, we can also consider regulating intestinal flora to alleviate anxiety symptoms. Study identifies dog breeds, physical traits that pose highest risk of biting children. New research at The Ohio State University College of Medicine and The Ohio State University Wexner Medical Center identifies dog breeds and physical traits that pose the highest risk of biting with severe injury. Doctors want parents of young children to use this information when deciding which dog to own. The study, published in the International Journal of Pediatric Otorhinolaryngology, explores the risks of dog bite injuries to the face in children and bite severity by breed, size and head structure. Researchers found pit bulls and mixed-breed dogs have the highest risk of biting and cause the most damage per bite. The same goes for dogs with wide and short heads weighing between 66 and 100 pounds. The purpose of this study was to evaluate dog bites in children, and we specifically looked at how breed relates to bite frequency and bite severity, said Dr. Garth Essig, lead author and otolaryngologist at Ohio State's Wexner Medical Center. Because mixed-breed dogs account for a significant portion of dog bites, and we often didn't know what type of dog was involved in these incidents, we looked at additional factors that may help predict bite tendency when breed is unknown, like weight and head shape. To assess bite severity, researchers reviewed 15 years of dog-related facial trauma cases from Nationwide Children's Hospital and the University of Virginia Health System. They looked at wound size, tissue tearing, bone fractures and other injuries severe enough to warrant consultation by a facial trauma and reconstructive surgeon and created a damage severity scale. Researchers also performed an extensive literature search from 1970 to current for dog bite papers that reported breed to determine relative risk of biting from a certain breed. This was combined with hospital data to determine relative risk of biting and average tissue damage of bite. There's an estimated 83 million owned dogs in the United States, and that number continues to climb, said Dr. Essig. We wanted to provide families with data to help them determine the risk to their children and inform them on which types of dogs do well in households with kids. According to the Centers for Disease Control and Prevention, 4.7 million people in the United States are bitten by dogs annually, and 20% of these victims require medical care for their injuries. Those who require treatment after dog bites are predominantly children ages 5 to 9 years. Young children are especially vulnerable to dog bites because they may not notice subtle signs that a dog may bite, said Dr. Charles L. Marafi, study co-author, associate professor of otolaryngology at Ohio State's College of Medicine and chief of otolaryngology at Nationwide Children's Hospital. We see everything from simple lacerations to injuries in which there's significant tissue loss that needs grafting or other reconstructive surgery. Dr. K. Craig Kent, Dean of the Ohio State University College of Medicine, said, "This research highlights a significant public health issue and provides a new decision-making framework for families considering dog ownership. The circumstances that cause a dog to bite vary and may be influenced by breed behavior tendencies and the behavior of the victim, parents and dog owner. Children imitate their parents," said Megan Herron, Associate Professor of Veterinary Clinical Services at Ohio State's College of Veterinary Medicine. Be a model for your child and avoid any confrontational or risky interactions that might trigger a fear or fear-aggression response if the child were to mimic it. This includes harsh reprimands, smacking, pushing off of furniture and forcibly taking away an item. Herron offers the following tips for dog owners. Most bites to children occur from a family dog when the dog is resting and the child approaches. Try to provide and encourage resting places away from where children run and play. Many bites to children occur even when an adult is in the room. If you can't devote your attention to the interactions between the dog and child, it may be best to have a physical barrier between them, such as a baby gate or crate for the dog. This is especially important for toddlers whose behaviors may be more erratic, unpredictable or frightening to a dog. Teach children to let resting dogs lie and to stay out of dog crates, beds and other resting places that are designated for the dog. If the dog's favorite spot is on the couch, put a towel or blanket down to clearly delineate the dog space versus child space. Children should not approach, touch or otherwise interact with dogs while they are eating. Provide quiet areas for dogs to eat away from areas where children run and play. Rawhides and other flavored chews should only be given when dogs are separated from child play areas. Teach children to find an adult if a dog takes one of their toys or snacks. Children should never attempt to retrieve these items themselves. Other researchers involved in this study were Dr. Cameron Sheehan, Dr. Shafali Ricky and Dr. J. Jared Christoffel. Owning a dog is influenced by our genetic makeup. A team of Swedish and British scientists have studied the heritability of dog ownership using information from 35,035 twin pairs from the Swedish Twin Registry. The new study suggests that genetic variation explains more than half of the variation in dog ownership, implying that the choice of getting a dog is heavily influenced by an individual's genetic makeup. Dogs were the first domesticated animal and have had a close relationship with humans for at least 15,000 years. Today, dogs are common pets in our society and are considered to increase the well-being and health of their owners. The team compared the genetic makeup of twins, using the Swedish Twin Registry, the largest of its kind in the world, with dog ownership. The results are published for the first time in scientific reports. The goal was to determine whether dog ownership has a heritable component. We were surprised to see that a person's genetic makeup appears to be a significant influence in whether they own a dog. As such, these findings have major implications in several different fields related to understanding dog-human interaction throughout history and in modern times. Although dogs and other pets are common household members across the globe, little is known how they impact our daily life and health. Perhaps some people have a higher innate propensity to care for a pet than others, says Tova Fall, lead author of the study, and professor in molecular epidemiology at the Department of Medical Sciences and the Science for Life Laboratory, Uppsala University. Carrie Westgarth, lecturer in human-animal interaction at the University of Liverpool and co-author of the study, adds, these findings are important as they suggest that supposed health benefits of owning a dog reported in some studies may be partly explained by different genetics of the people studied. Studying twins is a well-known method for disentangling the influences of environment and genes on our biology and behavior. Because identical twins share their entire genome, and non-identical twins on average share only half of the genetic variation, comparisons of the within-pair concordance of dog ownership between groups can reveal whether genetics play a role in owning a dog. The researchers found concordance rates of dog ownership to be much larger in identical twins than in non-identical ones, supporting the view that genetics indeed plays a major role in the choice of owning a dog. These kind of twin studies cannot tell us exactly which genes are involved, but at least demonstrate for the first time that genetics and environment play about equal roles in determining dog ownership. The next obvious step is to try to identify which genetic variants affect this choice and how they relate to personality traits and other factors such as allergy, says Patrick Magnuson, senior author of the study and associate professor in epidemiology at the Department of Medical Epidemiology and Biostatistics at Karolinska Institutet, Sweden and head of the Swedish Twin Registry. The study has major implications for understanding the deep and enigmatic history of dog domestication, says zooarchaeologist and co-author of the study Keith Dobney, chair of human paleoecology in the Department of Archaeology, Classics and Egyptology at the University of Liverpool. Decades of archaeological research have helped us construct a better picture of where and when dogs entered into the human world, but modern and ancient genetic data are now allowing us to directly explore why and how. Dietary cholesterol or egg consumption do not increase the risk of stroke. Finnish study finds. A new study from the University of Eastern Finland shows that a moderately high intake of dietary cholesterol or consumption of up to 1 egg per day is not associated with an elevated risk of stroke. Furthermore, no association was found in carriers of the APOE4 phenotype, which affects cholesterol metabolism and is remarkably common among the Finnish population. The findings were published in the American Journal of Clinical Nutrition. Findings from earlier studies addressing the association of dietary cholesterol or egg intake with the risk of stroke have been contradictory. Some studies have found an association between high dietary cholesterol intake and an increased risk of stroke, while others have associated the consumption of eggs, which are high in cholesterol, with a reduced risk of stroke. For most people, dietary cholesterol plays a very small role in affecting their serum cholesterol levels. However, in carriers of the apolipoprotein E phenotype 4, which significantly impacts cholesterol metabolism, the effect of dietary cholesterol on serum cholesterol levels is greater. In Finland, the prevalence of APOE4, which is a hereditary variant, is exceptionally high, with approximately 1/3 of the population presenting as carriers. Yet, research data on the association between a high intake of dietary cholesterol and the risk of stroke in this population group has not been available until now. The dietary habits of 1,950 men aged between 42 and 60 years with no baseline diagnosis of a cardiovascular disease were assessed at the onset the Kuopio Ischemic Heart Disease Risk Factor Study (KIHD) in 1984 to 1989 at the University of Eastern Finland. APOE phenotype data were available for 1,015 of the men participating in the study. Of those, 32% were known carriers of APOE4. During a follow-up of 21 years, 217 men were diagnosed with stroke. The study found that neither dietary cholesterol nor egg consumption was associated with the risk of stroke, not even in carriers of APOE4. The findings suggest that moderate cholesterol intake or daily egg consumption are not associated with the risk of stroke, even in persons who are genetically predisposed to a greater effect of dietary cholesterol on serum cholesterol levels. In the highest control group, the study participants had an average daily dietary cholesterol intake of 520 mg and they consumed an average of 1 egg per day, which means that the findings cannot be generalized beyond these levels. 1 egg contains approximately 200 mg of cholesterol. In this study, about a fourth of the total dietary cholesterol consumed came from eggs. Furthermore, the generalizability of this study is also weakened by the fact that the study population did not have a pre-existing cardiovascular disease at baseline and the size of the study population was relatively small. Therefore, the findings of the study should be verified in a larger cohort as well as in people with a pre-existing cardiovascular disease, who are currently advised to limit their intake of cholesterol and eggs.